Thank you very much. And I'm really sorry that I'm not there. Yeah, a bit of train chaos yesterday afternoon just proved to be a bit difficult to try and make it down to Oxford. So yeah, I'm really pleased. Therefore, it is a hybrid meeting, and I still get the opportunity to present. So yeah, today I was wanting to talk to you about our work on predictive phage therapy and the sort of proof of principle study that we've done, and then a bit more about the sort of future directions that we're taking this work, because we've talked a few times about the study, but I just wanted to summarize that again before talking about what we're doing now. So, so as we all know, there's several hurdles from where we are just now until we can actually get phage therapy into the clinic. A lot of them are centered around the regulation of using phage and the potential for commercial investment into phage therapy. We've also got so far still really a lack of kind of large scale clinical trials to sort of show proof of efficacy. And then we've got the issues around the production of phage and whether that will have to be through GMP facilities or whether there will be other routes for that. As academics, though, I think we are in a strong position to tackle one of the other major hurdles in bringing phage therapy to the clinic, and that's how we would actually select the best phage for treatment. And we can help to answer some of those questions and move that part of the work forward with some of the research that we are doing. Up in Edinburgh, our aim is to be able to establish phage therapy at Edinburgh Vet School for targeting, in the first instance anyway, canine UTIs. And so that's our kind of hope and vision for the future. And so the work we're doing just now is hopefully moving us in that direction. And as I said, one of the major issues that we have for phage therapy is how do you select the correct phage to give to the patient? And that's based around the specificity of phage to their target bacteria. And as we know, phages are often highly specific. And so we need to be able to find a way where we can efficiently identify the right phage for treatment in quite a timely manner. If we're wanting to maybe compete with antibiotics to treat an infection, we need to be able to come up with the phage relatively quickly to offer to the clinician. And so we set out to see if we could develop a smarter way to select phage for treatment and are based around this concept of predictive phage therapy. So although our work is focused on targeting canine UTIs, this obviously is very translatable to human UTIs, and so it is very much a One Health mission that we're undertaking here. So to use sort of this predictive phage therapy, we wanted to use machine learning and to test whether that is a useful tool for predicting phage activity on an infecting bacterial strain. And the type of machine learning that we've used is supervised machine learning, and that's where we take a labeled data set as our input data. We use that for training the models, the prediction models. We build these prediction models, and in our case, when we're looking at phage and bacterial interactions, our labeled data set is the interaction, the positive, the yes or the no. Can that phage infect that bacteria, alongside the bacterial DNA sequence? So it's a sort of genotype to phenotype prediction. And so we built models for each of our phage in our panel. And then once we had these models built, we could do some testing. So we would take some bacterial sequences back from the training set and put them back through the models and see how accurate they are at predicting whether the phage in question is going to be active or not on that bacterial strain. And then, of course, you can validate it with unseen bacterial strains as the final steps, which is what we've done. So ultimately, what we would do is we would get the bacterial DNA sequence from a clinical infection, put it into our models, and our models will predict which phage, or hopefully provide us with a list of phages that would be predicted and likely to be active to treat that infection. And so once you've built these interaction data sets and built these models, then you can hopefully bypass the manual testing, or certainly a lot of the manual screening. You probably still get to the point where you do manual a validation of the phage that have been selected in the lab, but it certainly cuts out a lot of the screening up until that point. So for us, as I said, our focus, our target was to look at canine UTIs. And so for us, when we wanted to build up this data set for training our models, we concentrated on UPEC bacteria, so uropathogenic E. coli. Most of these came from our canine patients, samples coming through the small animal hospital up at the vet school in Edinburgh. But we also had a set of clinical isolates that we got from the hospital to include the human aspect in this. Because we want the phage to be active in the bladder, in the urine, then we did all our testing in an artificial urine media. So we were trying to mimic the environment that we wanted the phages to work. So we took 300 bacterial, just over 300 bacterial strains. We had them all sequenced. So we had the whole genome sequence data, and we had a panel of 31 phage, and we tested using growth assays. So these were all done in 96 well plate growth assays, the 31 phage against the 314 bacterial strains. And we did each of these in triplicate. You can imagine this is quite a lot of interactions, a lot of data generated. And from this growth curve data, we scored the phage activity, and that was basically just done on an area under the curve calculation, where a high score was indicating a lot of bacterial growth, so really very limited phage activity. So a score of 100 basically means it grew the same as the control. A low score showed that we had good phage activity, and then these scores have just been translated onto with this color scale for this heat map for visualization. So here, where we've got the yellow colors, that means we've got some good phage activity, and the darker colors are where we have limited phage activity. And you can see from this that we have we've got a variety of broad spectrum and definitely narrow spectrum phage in our models, and that's important for how accurate the models ended up being, and I'll come on to that again in a bit. And for the sake of for some of the work that we've done, we drew a score at 60 to say that where the score was below 60, we would call that an active phage, and where it was above 60, we would call it an inactive phage. And so for this heat map, where the color starts to go that dark red and into the black, that's where our inactive phage are. So once we'd built our models, we ended up building 31 individual models. We wanted to validate that. So we took 10 completely new clinical isolates. Their DNA had sequenced and their interactions had never been anywhere near the training set, had them sequenced, and then used those sequences, put them through the models to predict which phage are likely to be active. We could then select which phage we wanted to put together to make a phage cocktail, and we could test them. As a comparison to that, we also had what we called our general cocktail. So that was just our best phage, our broadest range phage put together, sort of would be potentially an off-the-shelf cocktail. And then subsequently, we did the actual manual testing of our set of phage against these strains. And so then we could create a cocktail that would have been based on observed data. And we tested this both in our artificial urine media that we'd used for building up the database, but also sort of a final validation. We pulled some canine urine and tested these cocktails in the canine urine. And what we were really pleased to see from this was that our general cocktail, our off-the-shelf cocktail, performed the least well, as expected. There was no matching of phage to infecting bacteria. But actually, the cocktails we created using the observed data and the cocktails we created using the predicted data were very similar. So in activity and overall activity. So that was really good news that we can use these models to predict which phage could be useful for taking into the clinic. And this was, as I say, this was very much a proof of principle study, and we do appreciate it. It's the sort of first steps in moving this work along. And again, just from looking at our heat map, we can see straight away that we're missing phage. We don't have enough broad range phage to cover the UPEC collection that we looked at. There's around about somewhere between 15 and 20% of our isolates. We really would struggle to put a cocktail together. Even using observed data, we just don't have the phage that cover those strains. So we know we need to broaden our phages if we can, and we are in the process of doing that. But I have to say, at the moment, we're still struggling to find phage that really give us that activity across our whole set. Another thing we had to be mindful of was that when we built the models for the 31 phage, just less than half of them actually gave us really lovely, accurate, statistically accurate predictions. So I think it's roughly sort of about 13 or 14 of our models are useful in the context of what we're trying to do with them. And that's, I think, just is purely down to the balance of positive and negative phage activity. And we really need somewhere at least 15% activity across our strains before we have the data is good enough to build a model. So that's another thing we have to bear in mind for the future as we find new phage, that the phage not only have to cover our strains that we don't have phage for, but they have to have a broad enough activity across the strains so that we can build accurate models from them. So we know that we need to move this forward, we need to increase our capacity to build these interaction data sets. It's very labor intensive. The data set we've got there took the best part of 2 years to build using plate readers. We have increased our capacity now with a biolog machine that we can load multiple plates onto, and that's something that we're working up now. We also, like I said, we did this interaction set in artificial urine because that was the environment we wanted our phage to be able to work in. But of course, if we changed the environmental condition, our heat map would look very different. And we know if this was standard lab media, we'd have a lot more yellow on this map. Now, that might be useful for different E. coli infections and certainly useful data to have. So it's something we're considering, that going forward, how we build these interaction databases, what environmental conditions we use to make the data as broadly applicable as possible. And also to look at other people's phage sets. If we can get access to other phage libraries, then we can maybe be more efficient at building up these interaction databases to be useful clinically in the future. So the next steps for us just now are to really try and translate this into much more of a clinical pipeline, how we would envisage it would happen once we could roll this out clinically. And when we built the training data sets, we sent all our sequences away, all our strains away for sequencing. So that at best was a 4-week turnaround before we got that sequence data back. So obviously, as part of a clinical pipeline, that wouldn't be particularly useful. So we have just started using nanopore MinION sequencing, so we can get the DNA sequence direct from the sample. So directly from a urine sample, we can get plenty DNA to get MinION sequencing, do the genome assemblies, and put those into our machine learning predictions. And so we've been validating just now that we can use that slightly different sequence data and still have the same predictions, which we can, which has been really lovely to see that we've rerun some of our samples from before. So we've got both the short read and the long read sequences, and we get very similar results out. And so that's what we're optimizing at the moment. Ideally, this could all be done in one day. As soon as you get the urine sample in, we can extract the DNA and put it through the MinION sequencer. We're not doing that routinely just because of cost. At the moment, we're first checking we've actually got an E. coli in our sample before we load it onto the sequencer. But the vision would be in the future that as soon as the sample came in, we would just get that onto a sequencer and be able to predict which phage would be useful. And of course, you would also, with this kind of diagnostics, you can predict which antibiotics are likely to be useful as well, with the presence or absence of antibiotic resistance genes. And so that those 2 bits of information could be used in conjunction in determining the treatment plan for either the dog or the human in the future. And I think what we would see is that we would hopefully, in the vet pharmacy, we could have a selection of phage that had been produced to an acceptable standard, ready just to be taken out and put together as a cocktail to treat the animal, or as I say, human in the future. And so we're quite positive that we're not that far away from being able to do this as soon as the regulations and the things are in place, that we could actually then give the dogs the phage treatment. We're also looking to see if we can how we could commercialize this. Does this pipeline have a future in phage therapy? And we've been lucky at the Easterbush campus where Roslin Institute is situated. We've been part of a city region deal of data-driven innovation funding that's come through, and David Galley, our group leader, is involved with co-leading that, one of these hubs, which is a precision diagnostics and surveillance for One Health. And we really feel that we could maybe bring this phage data into this hub and use it as a way to be able to expand this and to do that, we've obviously got our own small collection of phage bacterial databases, but this would be something that you could bring together for other people that have got these databases, and then build up machine learning prediction models, and then potentially act as a coordinating hub in the future, where if a clinician can send a bacterial sample or the whole genome sequence, then we can run that through our models and advise on suitable phage from different providers of phage therapy. And so this obviously requires some kind of buy-in from the people that are able to then produce and distribute the phage to come in and see if this ability for us to predict which phage are likely to be useful can override some of the manual testing that has to be done at the moment. So if anybody is at all interested in what potential future we can have with commercializing this, maybe through our hub in Edinburgh, then please get in touch with us, because we'd be really keen to know if there was people that thought that this is something that they would like to be involved with. So we would, as I said, this is very much a proof of principle pilot study, and we know we can improve the accuracy of the prediction models, and that's going to be through understanding the biology. And one of the real delights of creating this huge interaction database is all of the biology that's just ripe for the picking that we can take from that. And for each individual phage and each individual bacteria, there's so much still to be discovered and understood about these interactions. If we take CHAP1, which is our broadest range phage, we know it uses OMP as its receptor. Because it's our broadest range phage, we had really nice balance of data for producing the models. So we have a high accuracy with the prediction for CHAP1, but there's still a small error rate. There's 7% are false negative and 11% are false positive. And by understanding why the model gets these wrong, we can maybe improve the models in the future. And I guess the two obvious things for the prediction is the receptor there, and what are the bacterial resistance mechanisms that are on the genome that play a role into this prediction. And but of course, with phage and bacterial interactions, it's not as simple as that. And we can have a look at the OMP sequence, and this is just our isolates, and actually a batch of 0157 isolates as well, that one of our students, Daniel Power, created a phylogeny based on just the relatedness of the OMP sequence. And then the blue sort of spikes coming out the end are the score. So a long spike is a score of 100, so no phage activity, and a small spike is where you've got better phage activity. And you can see there really is no relation between the OMP sequence and the phage being active or not. So it's as we obviously knew, we weren't expecting this to be anything different, but it's not as simple as a particular OMP sequence defines whether the phage is going to infect it or not. But what are the things that then do define it? And we really feel that to try and understand this, we need to peel back the layers. And we know that just having the receptor there is not enough, because the bacteria has got several different ways of being able to block phage absorption onto an available receptor. Of course, the receptors may be more available in one environment than they are in another, depending on the nutrient requirements and the metabolism, etc., of the bacterial cell. And then once you've kind of stripped that back, then you have the phage defense mechanisms and the presence or absence of them. So it becomes harder, I think, though, to predict the differences in gene expression at the environmental level. And I think that's where we're going down the road of investigating what sort of RNA-seq work we can do to really understand a bit more about the expression levels under different conditions, and whether that kind of data can also be put into our prediction models. And again, just carrying on with this theme that the biology is complex, a PhD student, Alba, in our group, former PhD student, she spent just about the whole of her PhD really trying to unpick the dynamics between one bacteria and one phage, and trying to understand that. Again, this was all done in the urine environment. And she took the strain EC958, so a well-known UTI strain. It's actually quite tricky to find phage that work well on it, but she found this phage lot 4 that worked really well. We got really good killing, But very quickly, the bacteria recovers. And interestingly, most of this recovery is not fixed mutants. The bacteria become sensitive again to the phage. And so she was really trying to understand that. But when we got some infected urine samples from a pig trial that we were doing, we noticed that when the phage worked a lot less well on this urine sample where the bacteria were already growing. And right enough, when we in vitro challenge the culture at 3 hours, we get very little killing. And so there's something obviously going on in the way the bacteria is presenting itself through the growth cycle. And we actually see this a really early OD, an OD of about 0.2, the bacteria start to become resistant. So alongside some other work and some other clues, we thought this could be due to capsule expression. And right enough, when we knock the capsule out, at 1 hour, it makes very little difference, maybe a little bit more sensitive. But at 3 hours, we do get a lot more killing. Bearing in mind, this is a log scale, so it doesn't look like a huge killing, but it's probably, it's pretty much a log of killing when we take the capsule mutant. So capsule expression is definitely playing a role in the phage activity. And can this be predicted? So the presence or absence of capsule genes is obviously important, but the type of capsule being expressed, can we predict that from the gene easily from the genome sequence? And when it's actually being expressed, because obviously at different points in the growth state of the bacteria, you're going to have more or less capsule. So if the capsule's there, then what the other genomic features that are present in the bacteria don't really matter if the phage can't get attached to the bacteria for a start. So these are all kind of things that we want to try and understand. And if we can understand which capsule each bacteria are likely to be expressing, we can try and make sure that we have phage in a cocktail that will be active on that capsule type. So that was just 2 small snippets of some of the information that really can be pulled apart. And hopefully, as I say, the more we understand about this, then hopefully the better our predictions can be by starting to consider these other aspects. And there is further aspects as well. Like I said, with this interaction data set, we have so much information to work with. And we know from a lovely paper from Rotem Sorik's group last year that the defense island repertoire of E. coli tends to go into 41 sort of hotspots. And so these are definitely parts of the bacterial genome that we want to include as important features for ML models, because that's likely to be where a lot of the defense genes will be situated. We haven't done anything yet with phage genome features, but obviously the anti-phage genes that phage carry will play a really important role in the prediction of whether that phage is likely to be effective on the bacterial cell. So that's something we're also considering. And even just going back, we've got a physics student starting just this month with us in collaboration, and his project's going to be based around really trying to understand a lot more of the bacteriophage dynamics, going back to look at the growth curves and seeing what we can tell in how the phage interacts with the bacteria at different stages of the growth curve, and how that could help our predictions as well. So in summary, we've shown that we can use machine learning models to develop phage cocktails quite successfully, and we obviously would like to expand that out to other infections. That rapid diagnostic sequencing will help us feed into this predictive pipeline, and hopefully a timescale that makes it clinically possible. We know we need to enhance our phage collection, and that it's really important that the more we understand of the biology, then we should hopefully be able to improve the machine learning models. And this is all work done in David Galley's group, and mainly Marianne Keith has been involved in all the generation of the data for the machine learning, and Anita Antonia did the machine learning models for us, and Alba spent a lot of time trying to understand the biology of EC958 as part of her PhD, which has been really insightful for us as to the complications that we need to address going forward. And of course, the Dogs Trust that have funded a lot of this work, we thank them as well. Thank you very much.